everyone at Sophion. It's great to be able to be here today to talk to you about the work that I have been doing. So, I'm a postdoc in the Robinson Lab at the University of Cambridge, where we work on excitable neuroendocrine cancers, and that's what I will be talking to you about today. So, neuroendocrine cancers are a subtype of cancer. They are characterized by their expression of neuroendocrine markers, such as ASCL1, and by their secretion of hormones. Primary neuroendocrine tumors are thought to arise from the neuroectoderm of healthy neuroendocrine tissue across the body, but it has also been observed that tumors of other origins are able to differentiate to acquire neuroendocrine characteristics, so there are multiple paths to arriving at a neuroendocrine phenotype. And I predominantly work on small cell lung cancer, which is a type of neuroendocrine cancer that represents a relatively small percentage of lung cancer overall, but it is extremely aggressive, highly metastatic, and can give rise to paraneoplastic syndromes, either due to its ectopic expression of ion channels or inappropriate secretion of hormones. And these types of cancers typically don't respond very well to traditional chemotherapy, so there's a real need for better understanding and better options, treatment options for patients. And a relatively, until recently, fairly understudied aspect of small cell lung cancer physiology is that these cancers are excitable, and this drives progression of disease. And this was shown recently in a paper from our lab, along with our collaborator Lian Li at the Crick Institute. And this paper looks at a variety of small cell lung cancer models, from mouse models and human cells and patient-derived xenograft tissues. And for many of the models in this paper, they produce two distinct cell populations. So a neuroendocrine type, non-adherent small cell population with high expression of ion channel genes, and a non-neuroendocrine cell population with much lower expression of ion channel genes, which are adherent and don't have the small cell morphology. And crucially, in the paper, they show that all of the neuroendocrine type small cell lung cancer cells exhibited excitability, whereas none of the non-neuroendocrine cell types did. And the paper then goes on to show that chemogenetic silencing of the firing is able to suppress metastasis in a small cell lung cancer mouse model, thereby linking the firing with the progression of disease. So this is the context and the background for the project that I am working on, and the aims of this are to identify the ion channels that are contributing to currents in human small cell lung cancer models and mouse small cell lung cancer cells, and then to look at these identified ion channels as a means of modulating the excitability of these cancer cells, in order to then look at the potential for modulating excitability on modulating the progression of disease. And in the lab, we are also working on refining a mathematical model for simulating our small cell lung cancer cells. But I'll be talking predominantly about the first two aims today. So the main cells that I work on, and the ones that I'll be talking about today, are a human small cell lung cancer line, the NCI-H889, and most of the work that I've done is on the mouse small cell lung cancer line AF1165, which was derived from a primary tumor from a double oncogenic mutant mouse model. So for both of these cell lines, they exhibit a small cell morphology and are non-adherent. And using whole cell patch clamp electrophysiology, we can see that they, yes, I'm not entirely sure why that has happened. But we can see that they both, well, we would be able to see, that they both produce robust outward and inward current indicative of voltage-gated potassium channels and voltage-gated sodium and potentially calcium channels. And using a current step protocol, we can evoke firing in both of these cell types. So if we compare the characteristics of the two cell types, there are significant differences. So for example, in the human small cell lung cancer line, we almost never see rebound firing after a period of hyperpolarization, whereas this is very prominent in the mouse line. And just to highlight that our measure of excitability is we take the difference of the peak evoked membrane potential from the steady state membrane potential. So essentially it is the amplitude of the spike overshoot. And there's also significant differences in the membrane properties between these cells. So part of what we're hoping to look at over the course of the project is whether we have species dependent differences or whether we just have significant differences between different models. So as part of the paper that I highlighted, a bulk RNA-seq dataset was generated and compared with published datasets. And this shows a high relative transcription of ion channel genes and the neuroendocrine cell types compared with the non-neuroendocrine cell types. Many of the channels are represented across lots of the model types, the different small cell lung cancer models. And I've highlighted a few here, and as you can see, there's quite a range of ion channels represented, which we were expecting. And many of these we would expect to be contributing to the excitability of the cells, and some of them have been implicated in cancers already. But obviously, RNA data does not necessarily mean functional expression of the channel. So a lot of the work that I have been doing is to validate whether we have functional expression of the channels that are indicated in the RNA-seq. There's not enough time today to go over all the channels that we've been looking at, so I will be focusing on the cation leak channels that we predict are contributing to our resting membrane potential. So one of the ion channel families that are highly represented across the small cell lung cancer models are the K2P family, which lots of people will be familiar with. K2P channels are outwardly rectifying potassium leak channels that have been shown to contribute to maintaining the resting membrane potential of cells, and altered K2P expression has been indicated in various types of cancer, but not really in with a consistent mechanism, so it is likely that the effects are probably cancer type specific. So the RNA-seq data suggests that the highest expressed K2P channel in the AF1165 cells is TREC1 with some TREC2, and antibody staining supports expression in the mouse and the human small cell lung cancer cells. So to look at this functionally, I've been using ML335, which is reported as a selective TREC activator, and we can see that 20 micromolar of this compound significantly increases the maximal outward current of the cells. And if we plot the conductance, we can see an increased slope factor indicating a reduced apparent voltage dependence of the overall outward current of the cells, which would be consistent with the mechanism of action of ML335, stabilizing the open conformation of the TREC channels and increasing this leak current characteristic. We also see that acute application of 20 micromolar of ML335 can reversibly hyperpolarize the membrane potential at rest, and this would support a role for the TREC channel in contributing to maintenance of the resting membrane potential. And interestingly, we also saw that slight acidification of the extracellular solution also produces a significant increase in outward. Sorry, I'm having my slides aren't really working. So sustained application of the compound is able to impair the excitability of the cells. So we see reduced rebound firing and reduced action potential firing, and hopefully we see that the membrane properties of the cells are also significantly impaired, significantly altered. So we see increase in rheobase and a decreased input resistance of the cells. And we are assuming that the impaired firing is due to the the effect on the membrane characteristics rather than altering the action potential generation of the cells. And so this is potentially a strategy for modulating the excitability of these cells and hopefully other small cell lung cancer cells in which K2B channels are expressed. Interestingly, we see that a slight acidification of the extracellular solution also gives a significant increase in outwardly rectifying current, and this may suggest TREC2 expression as well as probably TREC1 expression, as TREC1 would actually be inhibited by a lower extracellular pH, whereas the effect on TREC2 would be to give this increase in outwardly rectifying current. So sticking with the leak channels, we're also interested in the sodium leak current in our cells, and the RNA-seq data suggest that we have high expression of the sodium leak channel NALCN and the auxiliary proteins UNC79, UNC80, and FAM155A, which are necessary for current through NALCN. And staining supports the expression of NALCN in our mouse and human cells that I've shown you. The difficulty with NALCN is that there are not really any good compounds for targeting the channel. So in order to investigate the channel in our cells, what I have tried to do is block the other current and see what we have left, which I have done by using a potassium-free solution, sodium chloride based, with 1 micromolar TTX, as well as low calcium, low magnesium, and 5 millimolar cesium chloride, and a cesium-based intracellular solution. And that leaves us with a very small current, which obviously is still composed of several other channels. And so to try to isolate the NALCN current, we have used an NMDG-based extracellular solution. So NALCN cannot conduct the impermeant cation NMDG, and we can see that substituting that for sodium does reduce the current that we've already isolated. Using a voltage ramp protocol, we can see that it significantly and reversibly inhibits inward current, and that the NMDG solution is also able to reversibly hyperpolarize the membrane potential at rest. And if we subtract the current that we get in the NMDG solution from the initial current, we get our NMDG sensitive current, which we are hoping is a large proportion of this is NALCN. We can look at the existing literature to compare, and if we look at this paper from the PLAS lab, where they have overexpressed NALCN along with the necessary auxiliary proteins, we can see that in their voltage step protocol, which they've conducted under quite similar experimental conditions as us, their instantaneous current is similar to the current that we have identified as our NMDG sensitive current. So that is reassuring, but obviously we can't definitively say that this is an NALCN current. There are a few other things still that may be contributing. Trip channels, for example, would be the concern. So we were thinking about other aspects of the NALCN current that could be that we could try to replicate, and NALCN is rapidly and reversibly inhibited by extracellular calcium. So by decreasing the extracellular calcium concentration, we can see a reversible increase in the inward current, and also we can reversibly but sustainably depolarize the membrane potential at rest, and this would be consistent with reducing the calcium inhibition of this inward leak. Still, though, we cannot rule out many of the TRP channels. We do actually not have a very high expression of TRP channels in our RNA-seq, which is reassuring, but ultimately I think we're going to have to resort to genetic methods to really isolate the NALCN, but hopefully I've shown you that we are building a body of evidence that is at least consistent with NALCN expression. And the final thing that I wanted to show you is something that is quite new in the lab, and that is that we have been looking at 3D culture of our cells. So if you leave them to grow spontaneously, if you leave them to grow in solution, in suspension, they will spontaneously form these 3D spheroids, which we can plate back down and then patch. And we can see that they still have robust currents, and in fact, if we overlay the average 2D currents, it does look like we might be getting higher current in the 3D cultured cells. We can evoke firing still, and most interestingly, the 3D cultured cells are firing spontaneously. We do see this in the 2D cultured cells, but not as consistently and not at such a frequency. So far I've managed to keep the 3D cultures for about 2 weeks, and they are still pretty happy, still firing spontaneously. And if we compare the characteristics of the 3D cultured cells with the 2D cultured cells, it does look like there might be a difference in excitability, but I really am not ready to draw any strong conclusions until I've done a more robust study looking at the spheroids from independent 2D starting cultures and then matching these with 2D cultures from those same initial cultures. But what we have so far does seem exciting. So in summary, I have shown you one of our mouse small cell lung cancer lines and one of our human small cell lung cancer lines, that they both exhibit robust firing, but display different characteristics. I've shown you that between the RNA-seq and the staining and mostly the functional data, we have data consistent with the expression of leak channels such as TREC1 and 2 and possibly NALCN in our cells, and that our mouse small cell lung cancer cells are able to form 3D spheroid cultures that are spontaneously firing. So the project is still very much ongoing. We have quite a few next steps in the work. So we have a few more channels that we need to identify, that we need to validate, that have been indicated in the RNA-seq that would be important to regulating excitability. And then we want to go on to assess the impact of regulating excitability on the sort of cancer progression, so proliferation and motility of our cells. We want to further look at the 3D culture of the cells. And as I mentioned at the start, but I haven't been able to talk about, we are refining a mathematical model where we model in the individual conductances of the ion channels that we have identified in the cells to build a model that can simulate the firing, which so far we are able to simulate the firing fairly faithfully at the minute. So with that, I just have to thank my supervisor Hugh, my colleague Hao, and our collaborator Leanne. We don't have any lab photos, so you just have Cambridge to look at. Thanks again to the organisers, and thank you for listening.
Thank you very much. Fantastic talk. We're open for questions. Go ahead.
So great down here. So really nice talk. So you're going to be surprised, but I'm really curious about what happens if you put M335 on the cells for a while. What do you predict if you activate TREC1? What's going to happen to these cells? Are they going to proliferate or not proliferate?
I think we're not sure yet. I think based on results from the paper, where if you block the firing with TTX, you can impair colony formation, we might predict that if you impair the excitability with activation of TREC, you might see a similar thing. I think that's maybe what we would be hoping, is that you impair proliferation of the cells if we sort of, do you mean sort of cultured over a longer time, the
Yeah,
cells?
that's apart from acute.
Yes.
Because clearly, you're going to make them hyperpolarized.
Yes, yeah.
So the question is, if you keep them in that state for days,
Yeah.
are you going to block their proliferation?
I think that's what we would be hoping, based on what,
results?
yeah, I think, but yeah, we would have to do it, which is kind of the next step of the project.
Any additional questions?
Hi, Jodie, really nice talk. I was wondering if you had any thoughts on what would happen to protein expression in the 3D spheroids compared to your 2D culture.
Yeah, I think that would be really interesting to look at. I think, honestly, I'm not sure. I guess one theory is that maybe by culturing in 3D, we might be sort of increasing the maturity of the cancer progression. So maybe you would see the similar differences in expression that you see in progression of cancer, which from the paper there are certain, I'm not sure about sort of, they didn't really track the ion channel expression over time, but they looked at neuroendocrine markers over time and found some quite unexpected changes in sort of markers that were upregulated in the initial phases and then downregulated in more advanced stages of the cancer. So maybe we would see something like that with the 3D cultures if we're hoping that we're sort of pushing for a more like mature model. But honestly, yeah, it would be really interesting to look at, and I don't think we really know.
All right, thank you.
In synchrony with the excitation, do they release neurotransmitters that,
Yes, so we are also looking at, the theory is that as they fire, that promotes secretion of neurotransmitters, of hormones. We have actually done some capacitance measurements where we depolarize to stimulate firing, and we do see an increase in capacitance, which would be consistent with sort of transient vesicle vesicle incorporation into the membrane and release of neurotransmitters and hormones.
in the spheroid, are they releasing to the luminal or the outside?
Sorry, I didn't quite catch the question.
In the spheroids,
Mm-hmm.
where would they be releasing neurotransmitter to? Like just inside or outside, or is it hollow or?
Oh, in the 3D?
Yes.
Oh, they are, they're very dense. They're not, I mean, as far as we can tell, you can section through them and they are quite dense, and we can patch all the way down to sort of 2/3 into the spheroid and still patch cells. So, they're pretty dense.
Have you tried adding like TTX to the spheroids to see if it affects the growth of the spheroids over time?
No, so the spheroids are super new, but that is kind of what is exciting about them, is that we can culture them with compounds with TTX, and we can then measure the growth, and also we could then patch them at the end. So that is kind of the benefit of these, but we haven't, but that's kind of in the cards.
Okay, thank you very much.
Thank
And we'll
you.
move on to our next speaker.